Hey, Mr. P here. In this video, we're going to kind of extend our understanding of microscopy from a standpoint of how do we prepare specimens for observation with a microscope. We have to do a couple of things normally due to the fact that microbes are not naturally colored. They're kind of translucent, or just don't readily give off the contrast needed to differentiate between some of their internal structuring. So we've got to use staining, we've got to use smearing, we've got to use fixing, we've got to do some things to the specimen in order for it to, one, adhere to the slide so that we can move the slide around and actually observe the organism, and two, we have to specifically stain certain internal structures or certain external structures, or we need to differentiate one bacteria species from another. And so that requires some in-depth understanding of the structure of the cell in order for us to kind of apply our understanding of stains in order to prepare the specimens for observation with a light microscope. So let's jump in and get started. What does it mean to prepare a smear or a stain? Okay, staining is literally the act of putting dye on a bacteria or in a solution that we are going to bathe our bacteria in order to give contrast or emphasize a certain internal or external structure. Stains, as you'll see later on in this video, can be a variety of different types of chemicals, a variety of different types of dyes, and depending on the structure we want to emphasize, a different type of dye or a different type of staining technique needs to be employed in order to make that particular structure visible. Smearing is the act of putting a kind of spreading a thin film of bacterial material over the slide, usually then needing to be fixed, meaning it needs to be adhered, or it needs to adhere to the slide. Typically, we're going to fix our slides by running it through some heat from a Bunsen burner. That's called heat fixation or heat fixing. We will go through that technique later in the lab, but for the purposes of preparing us for lab, we need to make sure we understand how to properly stain and adhere or fix our bacteria species or bacterial material to our slide in order for us to observe it with a microscope. Okay, as you can see here, you can see what we call gram-positive bacteria as well as gram-negative bacteria. That probably doesn't mean much to you at this point because we haven't talked about the different morphological features of a gram-positive or gram-negative bacteria, but you can clearly see that each of them have a slightly different shape, maybe. They have a slightly different exterior surface, maybe, but they also, for the purposes of this video, have a substantially different color, and that's by design. Okay, later on in this video, we're going to talk about the gram stain technique. Gram staining is a way of differentiating between gram-positive and gram-negative bacteria. Gram-positive bacteria, based on the composition of their cell wall, is going to retain the purple color, and gram-negative bacteria, based on the composition of their cell wall, are going to not retain the purple and are going to be counterstained with red. Okay, this allows us to see side by side what the power or the purpose of staining is. It allows us to look not just at internal structuring or external structuring of the cell. It allows us to see very visibly the color that each of the cell types or each of the bacteria types will stain. So in order for us to understand staining and understand why certain cells stain a certain color, or even what stain to use in order to differentiate one bacteria structure from another, we need to kind of have a working knowledge of what stains are. And so stains are going to consist of a positive and negative ion. Okay, so they have both positive and negative ions. Based on whether it's a basic dye or an acidic dye, depends on which ion between the positive and negative is going to be colored, meaning we call that the chromophore. Okay, chromo obviously being a prefix that means color. And so when we institute a basic dye or construct a basic dye, we're actually going to put the pigment in as the cation, and we know the cation to be the positively charged ion. In an acidic dye, the chromophore is going to be an anion, which we know is the negative ion. Okay? Now, because bacteria are negatively charged, okay? Because bacteria are negatively charged, that dictates which stain we want to use in order to differentiate the structures we want to see. If we want to stain the actual bacterial cell, we're going to institute a basic dye, because that means the colored dye, or the actual chromophore, is a positively charged ion, which means that when we add that positively charged chromophore, it's going to bind to the negatively charged, or attract to the negatively charged bacterial cell. And it's going to actually stain the bacterial cell the color of the dye. Contrastly, if we want to stain the background, we're going to institute or employ an acidic dye, which means the chromophore is going to be an anion, or the negatively charged, and since the bacterial cell is negatively charged as well, it's going to repel the chromophore, and the chromophore isn't going to stain the cell. In fact, it will just stain the background. So, between these two, because the cell was negatively charged and the chromophore was positively charged, then the positive and negative charges attract to each other and they actually stick to each other. Okay, so the stain will stick and stain the cell. In this particular situation, because the cell was negatively charged, but the chromophore was a cation, or rather was an anion, and it too was negatively charged, the negative charge of the acid, okay, and the negative charge of the bacteria means that the acidic dye is going to be repelled by the bacterial cell and it will not adhere. And so all of the background material will be stained and the cell will not. We call that negative staining, okay, where the background is stained and the bacterial cells are not. So in this particular image, you can see that the negative staining allows us really to see the shape really clearly of the bacterial cell. It also would, if we zoomed in, allow us to see external structuring of the cell, whereas if we go to some of these previous pictures, like these, you can clearly see that the background isn't stained, but the cells are stained. Okay, so both of these stains would have been a basic dye, which allowed the dye to adhere to the acid or the negatively charged bacterial cells. But in the case of a particular negative stain situation, we would institute an acidic dye, and that would lead to negative staining. Okay, that is the big difference. So when we compare these two pictures, you can see that one image on the left is stained purple, and this image on the right is stained red. Again, we'll get into what that means specifically in the next chapter when we get into the actual internal structuring and internal anatomy of the prokaryote. But for the purposes of this kind of introductory chapter, we need to kind of understand what the vocabulary is. And so you will see that a simple stain is the use of a single dye that allows us to see clearly in full color the full cell, whether it be purple or red. Some of the simple stains you will see and use in lab in this particular class are methylene blue, carbofuchsin, crystal violet, and safranin. The methylene blue and safranin are both going to be discussed further in this lecture. When we talk about the gram stain, we'll go through a lot of gram staining techniques in this class in the actual lab. The gram stain and simple stain typically use a mordant, and a mordant is a chemical or a stain that typically is used to kind of maintain the stain or coat the stain so that it isn't readily washed off. It's kind of a fixative, or it's kind of an agent that makes the stain more permanent. And so a mordant, when you hear that term, it's just used to make the stain stick. Differential stains Okay, if simple stains are used to basically make the cell a single color, we can also use differential stains, which are used to distinguish between two different types of bacteria, or bacteria that have two different types of anatomy, or bacteria that maybe have or lack a specific structure. And so one of those differential stains is called the gram stain. Another one is called acid-fast stain. We will do both of these techniques in the lab. But we, like I said earlier, need to understand and have a good working understanding of the actual biology and knowledge of this concept before we get into the lab to make our lab time most productive. So, what is the gram stain? The gram stain is a technique that is used to differentiate, okay, it's a differential stain, it's used to differentiate gram-positive from gram-negative bacteria. Now, it isn't important that you understand what makes a bacteria gram-positive or what makes a bacteria gram-negative at this point. That will come next chapter. But for the purposes of this chapter, we need to understand what the gram stain technique is. And so the gram stain is a series of 4 steps that you can clearly see outlined in this particular diagram. The very first step is the application of crystal violet. That is a simple stain. It is a purple dye, and that causes every cell in your sample that you fixed to your slide to turn purple. Notice that I have rod-shaped bacteria and spherical-shaped bacteria. Again, we'll get into the actual nomenclature of those particular bacteria shapes later, but we've got rod-shaped bacteria and circular or spherical bacteria. Those are probably or most likely 2 completely different species of bacteria. But the very first step is the application of the crystal violet. That alone is not going to be conducive to total differentiation. It turns everything purple. And so then we apply the mordant. Like I said in the previous slide, the application of the mordant is supposed to make the stain more permanent. And so the mordant actually, because of the morphology of the cell wall of gram-positive bacteria, the mordant actually adheres the crystal violet more permanently into the spherical shaped cells in this particular diagram, and not based on morphological features of the rod-shaped bacteria. And so when we go to step 3, which is the alcohol wash, okay, we put basically ethanol onto our slide, and ethanol decolorizes our cells that the stain wasn't made permanent by the mordant. Okay, so to kind of back up, we put crystal violet on everything. Crystal violet turns everything purple. Our mordant adheres, because of some morphological features, which we'll discuss later, adheres the purple stain more permanently, to the spherical, it's called cocci, okay, to the coccus or the cocci bacteria in this particular diagram, and doesn't adhere the stain to the rod-shaped bacteria. When we get to our alcohol wash and our decolorization step, it will successfully decolorize the rod-shaped bacteria because the mordant wasn't successful at adhering that crystal violet to the bacillus in this particular situation. And then we counterstain with safranin, which again is a simple stain, but because safranin is successful in staining all cells red, it will stain all cells red. However, because the gram-positive bacteria were previously stained dark, the red actually kind of gets overshadowed and subdued in the gram-positive cocci bacteria, and so it just isn't showing up as red, even though the red stain is there. You can see at the end of our gram stain that we actually have some of our cells that are red, and we have some of the cells that are purple. You can now differentiate clearly between the purple cells and the red cells, and if you look at this picture, this shows you the exact same thing. You can clearly see that the rod bacteria in this actual microgram, or the microscope image, are gram-negative because they appear red after the conclusion of our gram stain. And we have coccus bacteria, which are showing up purple, and those are spherical, which gives it the designation of coccus, and they show up purple because they retain the purple crystal violet from the gram stain as well. So gram stain, like I said, is a differential stain technique. it allows us to differentiate the gram-positive from the gram-negative bacteria. Again, we will do that technique many times in lab. Now, if we differentiate our differential stain techniques from gram stain, now we go to acid-fast stain. In an acid-fast stain technique, we're going to do multi-step staining, but the stain is going to bind only to bacteria that have a waxy material in their cell walls. Okay, we call those acid-fast bacteria. And we use this technique to identify nocardia and mycobacterium, which are pathogenic. And the way that we do this technique is we use a primary stain called carbofusion. It causes all bacteria to turn red. Okay, notice that we have not differentiated at this step. Just like in our first step of the gram stain, the crystal violet turned all bacteria purple. That was not the differentiation step, or that step alone did not differentiate. This is the same way. Okay, we primarily stain our bacteria with carbofusion that turns all bacteria in our sample red. We then are going to decolorize with acid alcohol in this particular technique, which is going to remove the color from our non-acid fast bacteria, but it's going to not remove the color from our acid fast bacteria, meaning because we never remove the color, the acid fast bacteria will always retain the original stain, which was carbofusion, and so at the end they will always be red. When we counterstain after the decolorizing step with methylene blue, that's going to turn just the non-acid fast bacteria blue because we were successfully able to remove the color, which was red, from them, in particular during the decolorizing step of the acid-fast stain technique. Okay, so because we do this technique, step 1, 2, 3, and we were successfully able to stain everything red, and then the acid-fast bacteria retain that color, they'll be red at the end. Because we were able to remove the red stain and then counterstain with blue, the non-acid fast bacteria will be blue and the acid fast bacteria will be red. Again, we can distinguish between two different types of bacteria based on the acid fast stain technique. And the acid fast stain technique is literally used to kind of differentiate whether or not the cell has the waxy material around their cell wall or not. We're going to finish the lecture off with special staining. These three techniques are special stain techniques that are used to highlight one specific special morphological trait. A capsule stain is going to be used to actually distinguish bacteria that have a capsule present from bacteria that don't have a capsule present. The presence of a capsule in a bacterium indicates the microbe's virulence. Okay, I can't say it right now. Virulence. It basically is used to measure the degree to which that bacteria is pathogenic, and we know that pathogenic means that it causes disease. So typically, if a bacteria has a capsule present, it's more likely to be pathogenic and therefore more likely to cause disease. Since capsular material is water-soluble and may be easily dislodged during vigorous washing, and because it doesn't accept a stain very well, capsules look like halos around the microbe. So this almost looks like a negative staining technique where we're staining the background and only the little capsular material that doesn't retain stain shows up as this little kind of halo or zone of inhibition, if you will, that clearly indicates the presence of a capsule. Endospore staining is a special stain technique that is used to stain endospores. Endospores are resistant, dormant structures that reside inside of some cells that cannot be stained by ordinary methods. The way that we go about the endospore stain technique is that we use a primary stain of malachite green, and then we heat shock the cell to kind of cause the endospore to take on or to absorb that malachite green. We then wash the cells, we decolorize them with water, we counterstain them with safranin, which if you remember from our gram stain technique is the red stain. And when we go through this technique, spores appear green because of the malachite green, and then they have this little green dot within the red or pink cells. So the cells are stained pink from the safranin. The endospore is stained green from the malachite green, and if the cells have an endospore present, they look like a red cell with a green interior dot. The green interior dot being the endospore, and the cell that is red obviously is the red cell. Flagella stain is a stain technique that allows us to look specifically at flagella. Since flagella are so much thinner than other interior cellular structures, it requires the use of a different stain. Flagella stains use a mordant and carbonyl fusion to thicken the appearance of flagella, and that alone makes them easier and more visible under the light microscope. So typically, flagella alone won't take on the stain, and even if they took on the stain, they are so thin that it makes it very hard to view under a microscope. And so we have to kind of adhere a mordant to completely surround and thicken the structure of the flagella in order to make it thick enough for the light microscope to have enough resolving power to pick it up. Okay, those are all of the staining techniques. These are all of the different techniques that are used pretty frequently in the microbiology lab, which totally allows us to make different cellular components visible. It also allows us to differentiate one bacterium from another based on some morphological traits. And again, we're going to get into the actual anatomy of prokaryotes in the next chapter. But for now, make sure you know the different stain techniques, when they are employed, what they are employed, and how they are employed. We'll practice all of those techniques in the lab. Bring your questions to class. See ya.